Dear learners, I am Ramya, working as Assistant Professor in the Department of Biotechnology, St. Joseph's College of Arts and Science for Women, Hosur. We are going to learn about the scientist who made the significant contribution towards microbiology. The existence of microorganisms were speculative and are not based on any data or science. Till 17th century, microorganisms were neither proven nor observed by any people. Antony van Leeuwenhoek, Louis Pasteur, Alexander Fleming, Robert Koch, or various scientists experimented and discovered new ideas which are the significant contribution to the microbiology. Microscope, the basic instrument to find the microorganism, was discovered by Antony van Leeuwenhoek. Pasteurization by Louis Pasteur. Penicillin, a vaccine, was discovered by Alexander Fleming, and the relationship between microbe and disease was discovered by Robert Koch. Antony van Leeuwenhoek. He is called as father of microbiology, was the first person to observe microorganisms using a microscope of his own design and made one of the most important contributions to biology. Before Van Leeuwenhoek's discovery of microorganisms in 1675, it had been a mystery why grapes can be turned into wine, milk into cheese, and why food is spoiled. Van Leeuwenhoek did not make the connection between these processes and microorganisms, but using a microscope, he established there is a form of life which we cannot see in naked eye. Van Leeuwenhoek's discovery entered the long held belief that life spontaneously appeared from non living substances during the process of spoilage. This is the diagram of microscope. It has main thing, that is eyepiece, ocular lens, body tube, nose piece, diaphragm, mirror, base and stage. In the stage, above of the stage, the samples can be clipped in a slide where through eyepiece or ocular lens the sample can be viewed. Next is about Louis Pasteur. Louis Pasteur found the pasteurization process, which is a process of food product in which packaged and unpackaged foods are treated with mild heat up to 100 degrees Celsius to eliminate the microorganisms. Pasteurization destroys pathogens and enzymes that contribute to food spoilage, including vegetative bacteria, but most bacterial spores survives in this process. The process of pasteurization is named after him, Louis Pasteur, whose research in the 1860s demonstrated that thermal processing would deactivate unwanted microorganisms in wine too. Spoilage enzymes are also inactivated during pasteurization. Today, pasteurization is used in food industry for food preservation and food safety. To disprove the spontaneous theory of generation, Louis Pasteur in 1862 designed a special flask with a long neck called swan necked flask or S-shaped flask. He placed the nutrient medium and boiled it to ensure sterilization. Though the flask was kept open, no growth was seen in the medium due to long neck. Later, when the neck of the flask was cut open, the nutrient broth was exposed to air. Soon, microbial contamination was there in the nutrient medium. Alexander Fleming. Alexander Fleming is known for vaccine called penicillin. He identified from mold bred to infected wounds. But it was not until 1928 that penicillin, the first true antibiotic, was discovered by him, the professor of bacteriology. 1928, Fleming began to sort through petri dishes containing colonies of Staphylococcus bacteria that causes boils, sore throats, and abscesses. He noticed something unusual on one dish. It was dotted with colonies and save for one area where a blob of mold was growing. Then the zone of inhibition immediately around the mold, later identified as a rare strain of Penicillium notatum, secreted substances that inhibits the bacterial growth. Robert Koch. He is a German physician and microbiologist. He discovered the relationship between microbe and disease. Koch postulated few theories. Number one, the microorganisms must be found in abundance in all organisms suffering from the disease but should not be found in healthy organisms. Number two, the microorganisms must be isolated from a diseased organisms and grown in pure culture. It can be grown in pure culture by doing the subculture, and the microorganisms can be isolated from a diseased organism where we can get the pure microorganism culture. The cultured microorganisms should cause disease when introduced into healthy organism. It means that the cultured microorganisms can cause infection even to the healthy organism. The microorganism must be re-isolated from an inoculated diseased experimental host and identified as being identical to the original specific causative agent. Today's question is, who is called as father of microbiology? And the other question is, who discovered pasteurization process? Thank you.